Hello everyone. So I'm back with the lesson Fundamental Unit of Life, and in this chapter this is our third lesson, third video together, right? So today we'll be going in depth about nucleus and cytoplasm. These are two parts of the cell and we'll be looking at them in detail. So we'll be seeing their structure, how they look under the microscope. We'll learn about its function, right? So lots of exciting stuff ahead. So what are we waiting for? Let's dive right in. So remember the experiments that we already looked at virtually. So these are the results from those experiments. That is the cells of the onion peel, which has been stained with safranin, that is the pink one, and the cells of our cheeks, so which has been stained by methylene blue. So in this, the dense region in every cell, the spherical dense region in every cell is the nucleus, right? So nucleus, what are they? When cells are observed under the microscope, the darkly stained spherical or oval structure near the center of each cell are called nuclei, which is plural, and the singular word for that is nucleus, right? So there are a lot of terms inside nucleus that if we get confused, it will be a big problem, right? So in order for us to get the concepts right, let's just first diagrammatically see what the nucleus has. So suppose this rectangle that I'm drawing is a cell. Then near the center we have a darkly stained, a dense spherical area which is nothing but the nucleus. That's the cell, that's the nucleus. Now if we just take the nucleus and magnify it even more, what we get to see is this. A double layered envelope or membrane that has really small pores in between. That's the double layered membrane, which is called as nuclear membrane. Since that membrane belongs to the nucleus, we call it nuclear membrane. And remember that it is a double layer. Which has many nuclear pores or small holes. Now inside the nucleus also, there is another darkly stained area that is mostly spherical, and that is called as nucleolus. Now nucleolus is the part that is responsible for synthesis of many types of proteins. And inside it, you find the genetic material, right? The material that has the information for what is to be transferred from the parents to the offspring in the form of chromatin. Now let's look at what chromatin is in detail. So it is seen as thread-like fibrous material. Thread-like. And it is nothing but tightly packed DNA. Tightly coiled or tightly packed DNA. Now we are all familiar with what DNA is, right? And we'll look into it in detail. Now why do we need to pack DNA? Because in humans itself, average length of a DNA is 5 feet. Right? It's almost the height for average grown up. So during the rest phase, this DNA is tightly coiled in the form of thread-like or fibrous chromatin. Now, at the time of cell division, these chromatin condenses into, that means it becomes even more closer and more coiled and more tight. Right? It condenses into what are called as chromosomes. So what is the difference? Chromatin is when DNA is tightly coiled in the form of thread-like structures during rest phase of a cell. Whereas what is chromosome? During the time of cell division, the chromatin, when it condenses into chromosomes. So what is the difference between the two? Chromatin fibers are thinner and they are thread-like. We have already learned that it's nothing but tightly packed DNA. Now what are chromosomes? How are they different from chromatin? They are thicker or denser since it's even more tightly packed, and they are rod-like. See, at some stages they look like this, rod-like. And then chromosomes are also nothing but tightly packed DNA, but actually extremely tightly packed DNA. That is, chromosome is also extremely tightly packed DNA. It's tightly packed DNA, but it's the extreme. It's as tight as DNA can be packed, right? Not just DNA, we have to remember that in both chromosomes and chromatin, the DNA is packed around proteins, right? So that's what retains its structure. Now, we all are familiar with DNA, how it looks, right? We have seen in almost so many hospital posters. We'll see it in movies and series that are, crime thrillers and all that. We are familiar. So, it has a structure which we call as double helical, and it's a large molecule. Now, a segment of DNA, not just any segment, but it has to be a functional segment. That means that segment has to be responsible for a trait like the eye color or the hair color or the height, some trait like that. So such a functional segment of DNA is called as gene, right? So this is the basic overview of what we are going to learn about the nucleus. Now, let us look at each of these in detail, and let us look how all of these look under the microscope. Let us see how they function, what are their functions. So, let's go into the details of all of this, right? So, first let's take the nucleus and see its structure. As we already saw, it has a double layered covering called nuclear membrane that has nuclear pores. So, I don't know if you can see it clearly here. This is the microscopic image of the nucleus. And this is the double membrane. And in between, you can see small pores. We'll see even more pictures, so that will be clear. Then what else does this have? Nucleus houses a small, dense, spherical structure called as nucleolus. Right? Well, how does it help? It helps in making or synthesizing some of the proteins. And nucleus also contains information for inheritance of characters of parents to the next generation. So that means, how is this information present? In the form of DNA that is coiled and packed into chromatin. Here you can see the chromatin fibers, okay? So into chromatin or chromosomes. And another part of the nucleus is the nucleoplasm, which just like the cell is filled with fluid-like area, a lighter area that has fluid called cytoplasmic fluid. Even the nucleus also has a fluid matrix and it's called as the nucleoplasm. Okay. Now there are some important terms that we all will be asked in examinations frequently. So let's look at them one by one. So inside the nucleus, about the contents of the nucleus, there are a few important terms that we all need to be clear about, and these are important because obviously they are often frequently asked in the examinations, right? So let's first look at DNA. Now DNA stands for deoxyribonucleic acid, right? Now it is a double helical molecule. That means what is the structure of the molecule? Double helical, okay? And it is a large molecule, as we have already discussed. So a double helical large molecule that contains the information necessary for constructing and organizing a cell. That means the information contained in the DNA tells a cell about how it should develop into, how it should be constructed, right? So from a larger perspective, that means if every cell is being told by the DNA how to develop into and how to function, it means from a larger perspective, it actually carries the instruction an organism in itself needs to develop, live and reproduce. That means the DNA along with the environment are responsible for how we develop, live and reproduce. Okay. These instructions are passed from parents to children. So in a way, how is the information passed? Through proteins, right? So in a way, it's the recipe book that holds the instruction for making all the proteins in our bodies, right? Now gene, what is it? So they are functional segments of a DNA. Not just any segment, but functional. They need to function. What is the function of the gene? To code for or to be responsible for a particular trait, particular character. So a gene codes for a particular trait like eye color or hair color or the height of an individual, right? So now chromatin. What is chromatin? So here is another picture. First is a lighter picture, that is a lower magnified picture of how the nucleus looks, and the second one is even more magnified, right? So you can see the nucleolus, the nucleoplasm and the chromatin. Now into the types of chromatin, you'll learn in the higher classes. So what is chromatin? It's nothing but the entangled mass. So you remember how I drew the chromatin? Like this, right? So it is the entangled mass of thread-like structures inside the nucleus, which contains tightly packed DNA around proteins. So when the cell is about to divide, this chromatin material gets organized into chromosomes. Why? Can you think why this organization is needed? Why can't chromatin be just like that? Because during a cell division, all these genes need to be correctly divided, right? So for that to take place without any confusion, it needs to get more organized. It needs to get more condensed and more manageable. And what is that form? It is chromosome. So when the cells are about to divide, chromosomes are visible inside the nucleus as rod-shaped structures. So you can get a magnified diagrammatic representation of a chromosome. So you can see it's in the picture that it is nothing but DNA that is very, very tightly coiled, right? So, and packed. So it contains very tightly packaged DNA around proteins, right? Now, what are the functions of the nucleus? So, obviously, because all this is happening, that is the chromatin is getting divided, organized into chromosome. So during cell division. So all this in itself implies that it helps in cellular reproduction to take place without any confusion, right? So it plays the central role. That means it plays the most important role in cellular reproduction. How do cells reproduce? By cell division. That means one cell divides into two, right? And it also plays a crucial part along with the environment in determining the way the cells will develop and what form the cell will take at maturity, right? That is nothing but cell differentiation, the process through which a cell develops and organizes itself to form its final structure. That is the way the cell will develop and the form that it will take at maturity. That whole process is called cell differentiation. That means a basic cell differentiates itself into what it's supposed to do and how it's supposed to look like. That means how a normal cell gets divided and forms into maybe a nerve cell. That process is called as cell differentiation, where it specializes, right? So for the cell differentiation also, nucleus plays an important part. How? By directing the chemical activities of the cell accordingly. So it plays a central role in cellular reproduction, and it also plays a crucial part along with environment in cell differentiation. That is in determining the way the cell will develop and what form it will take at maturity. How? By directing the chemical activities of the cell. So you can see another diagrammatic representation of the nucleus in three-dimensional view you can get in this picture. So you see it's a spherical structure and it has a double membrane and there are nuclear pores. Now what is the function of these pores? Can you guess, or can you just make out from what all we have learned? The function of nuclear pore is nothing but to act as doorways, right? So all these functions of the nucleus can only happen if there is a communication between the nucleus and the cell, rest of the cell. And this communication, how does it happen? By transfer of chemicals and other structures, other substances. So for this transfer, there need to be door. If it is completely closed, it can't take place. So these nuclear pores acts as doorways for substances to travel between the cytoplasm and the nucleus. Then you have a dense spherical structure called as nucleolus inside the nucleus, and the nucleoplasm, which is the fluid matrix inside the nucleus. Then here we can see the DNA in the form of chromatin, right? Thread-like, entangled mass of thread-like structures. That was about the nucleus, and now let us move on to the next topic, cytoplasm. So to see what cytoplasm looks under the microscope, we have already seen. So I'm taking what you know already, right? I'm building on that. So we have learned that inside the cells. So that was about the nucleus. Now let's move on to the next topic in this class, that is the cytoplasm, right? So I'm again coming back to what you already know, right? I'm taking the strong foundation of what you have seen many times, what you are familiar with, what you know, and let's build the rest of the lesson on that strong foundation, okay? So here we have seen plenty of times the onion peel slide and the cheek cell slide, right? So in these we have spotted the nucleus already as the dense region inside each cell. Now there is also a lightly stained region in every cell, and that lightly stained region is nothing but the cytoplasm, right? So it's the large region of each cell that lies between the cell membrane and the nucleus. Inside a cell, it's the large region that lies between the cell membrane and the nucleus. And it takes up very little stain when viewed under the microscope. What is another characteristic of the cytoplasm? It houses the various cell organelles. All these various cell organelles we'll be looking into in our next lesson. We'll start with it in our next lesson. So for now, just the nucleus and cytoplasm, right? And until now, what have we learned? How does the cell look like, basically? So we have learned there is a cell membrane and there is a cytoplasm inside which there is a densely stained spherical structure called the nucleus. And how is the genetic material organized inside the nucleus? What did we just learn now? That there is a nuclear membrane that envelops this nucleus. There is a nucleolus and the genetic material is organized, is available in the form of DNA that is organized as chromatin or chromosome. Now this organization, which is neat, right? It's got a clearly defined nucleus which is separate from the cytoplasm because it has got its own membrane. This kind of arrangement is seen in highly evolved organisms like our cells. So such organisms are called as eukaryotes. So in eukaryotes, there is a clear defined nucleus, and inside that the genetic material is neatly organized, right? Now there are also some other types of organisms called as, which are more primitive, and therefore their organization is also not so advanced. So in such organisms, how is the genetic material found? So inside the cell, the genetic material is loosely, it just lies in a poorly defined area loosely, right? So here you have the chromosome and the plasmid, both are genetic material in different forms. And such a poorly defined area where only the genetic material lies as nucleic acids only, and such a poorly defined area is not called as nucleoid. There is no well-defined nucleus which is enclosed by its own membrane, but just a region in which the genetic material is present. So such organisms are called as prokaryotes. Pro meaning primitive, and karyotes comes from the word karyon, which means the nucleus. So prokaryotes, right? Now let's look at the major differences between a prokaryotic cell and a eukaryotic cell in detail. Let's make it even more clear and crisp. Okay. So this is also a frequently asked question. So with respect to size, a prokaryotic cell is generally small, that is 1 to 10 micrometers. Now to get an idea about what this looks like, let's look here in this picture. So this picture tells us how big is 1 micrometer. Now here human hair is shown. One single strand of human hair has a diameter of 40 to 20 micrometers, right? So one single strand of our hair looks so thin, so small for the diameter of it looks so small for us, right? So that is 40 to 20 micrometers. The size of our red blood cell is 5 to 10 micrometers. And that of a spider web silk thread is 2 to 3 micrometer. Now, a bacteria is 1 micrometer. Right? So, here, the general size of a prokaryotic cell is usually small from 1 to 10 micrometer, and a eukaryotic cell is generally larger. That means 5 to 100 micrometers. Now, how is the nuclear region in both these kind of cells? In prokaryotic cell, the nuclear region, as we already saw, is poorly defined and it contains only nucleic acids. This poorly defined nuclear region containing only nucleic acid are called a nucleoid, right? Now, in eukaryotic cell, the nuclear region is well defined and it is surrounded by a nuclear membrane. How are the chromosomes found in prokaryotic cell? Only single chromosome is present, whereas in eukaryotic cell, there are more than one. And finally, about the cell organelles. Now, the cell organelles in prokaryotic cell are not bound by any membrane, whereas in eukaryotic cell, all the cell organelles are bound by membrane. Now, why is membrane so important? Right? So, we can see the significance of membrane in certain organisms that are special or peculiar. Now, by now, I'm sure all of us are familiar with this picture. I think even small kids are familiar with this picture. Can you guess what it is? It is the picture of a coronavirus, right? So, viruses show us the importance of membranes. So, viruses are peculiar organisms in such a way that they lack all membranes. There are absolutely no membranes in a virus of its own. So, as a result, that means as a result of the absence of membranes, these viruses do not show any characteristic of a living organism. That means it doesn't show any characteristic of life until they enter a living body and use its cell machinery to multiply. That means until and unless a virus gets into a living body, it does not show any characteristic of life. Once it enters a living body, only then it uses the cell machinery of that living organism and it multiplies. Only then it starts showing the characters of a living organism. Now here are 3 pictures of different organisms, right? Credits to Khan Academy over here. I borrowed this from one of their questions. So in these 3 organisms, can you spot which of these is prokaryote and which eukaryote? Right. So here we can see in this picture there is a clearly defined nucleus. Whereas here also we can see a nucleus. What about here? There is just DNA over here, nucleic acid. Genetic material is present in a poorly defined area as only nucleic acid, right? So this is what these organisms are, eukaryotes, and this organism is a prokaryote, right? So I hope we are clear with our concepts here. Let's do a quick review about what all we learned today. First of all, we learned in detail about the nucleus and what it has, right? The structure of the nucleus. It has double membrane. It has nuclear pores in the membrane. It has fluid matrix called nucleoplasm. It has a dense region that is nucleolus, and that was this. And there is genetic material, that is DNA, present in various forms, that is as thread-like chromatin, which gets organized, condenses into rod-like chromosomes during cell division, right? So, nucleus is the darkly stained, and cytoplasm also we learned. Cytoplasm is the lightly stained part when observed under the microscope. So, inside the nucleus, about the genetic material also, we learned in detail what a DNA is, what a gene is, what chromosome is, what chromatin is. Next, we learned about the cytoplasm, that it houses cell organelles and it lies between the nucleus and the cell membrane, and under the microscope, it is the large, lightly stained area inside a cell, right? Then we looked into how this genetic material is organized in different types of organisms. We learned that there are prokaryotes and eukaryotes. In this picture, the second picture, organism A, what is it? Is it a prokaryote or a eukaryote? So, genetic material is present only as nucleic acid. So, it's a prokaryote. We have seen a more clear picture of a prokaryotic cell, which is this, right? So, it's got a poorly defined nuclear region called nucleoid, that in which genetic material is only present as nucleic acid. And we also learned that in eukaryotes, the genetic material is present inside a well-defined nucleus, okay, that has its own membrane. We learned the major differences also between prokaryotes and eukaryotes. So this was our lesson for today. Thank you very much for watching. Please subscribe to my channel and share these videos with your friends, teachers and family. Please like this video if you like my work and comment your feedback. Thank you very much. See you next time.